All right, I think I'll kick it off. So welcome to the Linda Loring Nature Foundation Virtual Science Pub. My name is Seth Engelborg. I work as the naturalist educator and program manager at the Linda Loring Nature Foundation, and we're excited to host this event, Virtual Science Pub. This is only the second time ever we've run a virtual science pub, but we've been running a regular science pub at a local restaurant or bar on Nantucket for the past 6 years. Today's speaker is Dr. Sarah Bois, and she was the person who brought Science Pub to Nantucket, so we thank her for that. The model is very much common in university towns. It's kind of trying to get research talks outside of an academic setting and trying to bring them into a more of an informal environment, like a bar, so that people can connect with science and nature on more of a personal level. Just a few logistics about Zoom. We are doing a recorded webinar. So the only people who have speaking privileges and video privileges during this talk are myself as the host and Sarah as the speaker. If you have a question that you would like to address generally, you can put it in the chat and I'll be fielding those questions. If you have a question related to the presentation, you can ask that in the Q&A. And at the end of the presentation that Sarah gives, I will be fielding directing questions from the Q&A to Sarah as well. In the chat, I have posted the link to this webinar, the meeting ID, and the password. So if any of you are on and have friends or family members who are having trouble joining, you can forward that information to them directly. We'd also like to thank the Saltbox Tavern and Table. That's a local restaurant and bar in Nantucket that had been hosting this event for the past 2 years. And even though we're not able to do an in-person science pub right now, we really appreciate their support. And we urge you to support them in the future for all the they've been helpful in being a venue for us. So we hope you support them in the future. And with that, Sarah, is there anything I missed?
I don't think so. Oh, the only thing we wanted to say was if you were online for Jay's presentation last week from the Mariah Mitchell Association, he, even if you weren't here last week, if you're interested in his talk, we did record it. We're just working on the best platform for us to upload it because it is quite long. You know, it's almost an hour with the questions. So, and this being our first time doing a virtual science pub in general, you know, we're just working on getting that up. So hopefully that'll be up in the next few days. And then the recording from this talk will be up as well. So keep an eye out for that if you're interested.
Yeah, and just to add to that, if you're wondering how you can access those videos, we'll put information on our website and our Facebook page to give you instructions about how to access the video from last week and today. So stay posted. Other than that, I think it's time to kick off today's Science Pub. Our speaker is Dr. Sarah Bois from the Linda Loring Nature Foundation. She is our Director of Research and Education. She'll be giving a talk titled LLNF BioBlitz for Nantucket using iNaturalist. And we really look forward to seeing what she has to say. So thank everybody that's on the webinar as an attendee. And with that, I'll hand it off to Sarah.
Great.
Sarah, I'm just gonna follow your video, turn my video and my mic off, and I'll let you have the floor.
Great. Thanks, everybody. And just bear with us with these logistics. I'm gonna share my screen so that you will be able to see our presentation. All right, and I'm going to put the, sorry, just putting it into, there you go. All right, so thank you all for being here tonight or this afternoon. you know, before I get into the talk, I just wanted to mention that, Science Pub is usually once a month. And last week when Jay presented, he was pre-scheduled to be our last Science Pub of the season. We usually end in April, right before Daffodil Weekend here on Nantucket. And we thought that in light of the stay-at-home orders and, us trying to bring different types of programming to the community, we thought that continuing Science Pub once a week with additional speakers would be something that we would try for April and see how it goes. And so that's one other thing that if you have feedback about it in general and suggestions for speakers, that would be great. At the end of the talk, we'll mention next week's speaker as well. So once again, I'm Sarah Bois. I'm the Director of Research and Education at the Linda Loring Nature Foundation, and I'm going to be talking today about the LLNF BioBlitz and using iNaturalist. And so just briefly, this is the outline for what we'll be talking about today. So I'm going to talk generally about what a BioBlitz is. Some of this may be very familiar to people on the webinar, but some of it might be really new. So I just want us to be all on the same page. The goals of our LLNF BioBlitz and why we started it, what iNaturalist is as a platform, and then a how-to. So how do we get started as individuals, as community members, as families that want to get started with iNaturalist. And then this is a very much an ongoing process, project. A lot of times when we're presenting at Science Pub, we're presenting a project that's already complete and we're showing results. So we're going to talk about where we're at now, mid-process for the BioBlitz, and then what's next for the BioBlitz, and then additional resources. So there's lots of additional resources that you can check out on your own. So we'll be going over all of these things. So first up, just to make sure we're on the same page, is what is biodiversity? So I'm going to be using the term biodiversity a lot. We're talking about the variety of life forms that can be found in any particular ecosystem. Because this is focused on Nantucket as a BioBlitz, we're going to be talking about the Nantucket ecosystem, but people I recognize might be visiting or being on this webinar from many different places. You can think about the system where you live. And we're talking specifically about life forms. There's lots of cool things in nature that aren't alive, but we're talking about live things right now. So what is a BioBlitz? So traditionally, a BioBlitz is a survey for that biodiversity. So it's usually an intensive survey over a defined area. So like a Nantucket BioBlitz would be an intense survey over Nantucket. And usually it's over a short period of time. So for example, when I lived in Connecticut, there was a Connecticut State BioBlitz that was run by the University of Connecticut and the Connecticut Natural History Museum. And it was specific areas over a weekend or maybe even a week to 3 days. And scientists get together with community members, families, school groups. And it's a way that local naturalists and scientists can educate the public about the biodiversity around them. Usually they're very well planned out. There's lots of organized group activities. And like this picture on the bottom from the Delaware Nature Society, it's usually big groups of people focused on an area or a specific taxa. Like in this case, there was an insect survey, for example. However, we're not really able to do that right now. So our BioBlitz looks a little different. So we decided to start the Linda Loring Nature Foundation BioBlitz sometime around mid-March. Like a lot of conservation organizations and education organizations, we were thinking during stay-at-home orders when we had to practice social and physical distancing, we were really thinking about how we could pivot what we do as an organization and continue to provide programming. So part of our mission is to bring science and nature to our community. And our community is at home right now. So that's one of the reasons we wanted to start this project. But also, like the pictures I have here on the right side of my screen, these are pictures of what we're doing every day. We're taking walks every day. I kind of joke this is my series of me walking behind my family. And so people are getting out more and more on a daily basis, exploring new trails, exploring new properties they've never been to before, and maybe taking walks at a time of year that they're not used to exploring. On Nantucket, people are really used to going to the beach in the summer all the time. And this is an opportunity for us to learn more about our island. So some of the goals for this were for people to learn more about the biodiversity of the local environment. So it's a way for people to learn about it, but us to also gather that data. And it's also a way that we can help people gain an appreciation for local plants and animals as we learn more about what's around us. And also an understanding of the timing of natural events. So the timing of natural events is called phenology, and that's something that I study. But you know, as we start this process in March and we're continuing into May, this is a perfect time of year really to be out and about and looking and seeing what's happening, because things are changing, right? So thinking about when things are leafing out, when birds are arriving back, when things are coming out of hibernation. And so we're kind of really getting the lay of the land of what's happening now. And then it's also a way for us to stay connected when we're physically apart. So it's a way by, you know, the application that we have and the way that we're doing this is such that people are sharing the information that they're gathering, or the biodiversity observations they're making. They're sharing that with the community of people who are also interested. So it's a way for people to share their love of nature and the places that they're visiting with the rest of the community. So how are we doing that? So because we can't all be together and take nature walks together and keep lists, we're using a platform called iNaturalist. So iNaturalist is a citizen science program that was started in the late 2000s. It was actually started as a graduate student project and has flourished to be a global biodiversity database. So currently now it's a project that is a combination of the California Academy of Sciences and National Geographic. And it collects biodiversity information around the world. And so this picture right now is of the main page on their website. So if you haven't signed up before and you go to sign up, right there when you first open it up, next to this beautiful mantid picture, you'll see where you can sign up for an account. And it's all free. And it's also a citizen science platform, which is perfect because Citizen Science month is April. And since we're in April, it's a perfect time to start a new citizen science project. So for the LLNF BioBlitz, working with iNaturalist, these are the 3 basic steps of how it works. You see something, you record an observation, and in that case, it's a photograph of what you see. You share it with your fellow naturalists via the iNaturalist platform, and then there's an opportunity to discuss findings. And so we'll go into more detail about how that works, but it's actually quite simple. And using iNaturalist, it works on all of your devices. So if you have a device where you can take a picture, iNaturalist will work. And even if you don't want to use a smartphone or a tablet, if you are out and about in nature and you have a digital camera, you can take photos with your digital camera and upload them later onto a desktop or a laptop computer. And all of these data points, these photographs you take, are data points that contribute broadly to science. And I'll talk a little bit more about that in a minute. But not only does the Linda Loring Nature Foundation get that information for our BioBlitz, but it goes into iNaturalist, which then goes into the global biodiversity database. And research grade observations, which I'll talk about what that means in a minute, are then used into or put into the Global Biodiversity Information Facility, also known as GBIF. And lots of scientists use GBIF species information for lots of different research projects. All you have to do is take a picture and you can support science in all these different ways. And the other thing to remember is that iNaturalist, being a citizen science project, has open data for use and reuse. So once you make an observation and upload it into iNaturalist, like I said, it's available for the Linda Loring Nature Foundation, but it's also available for other scientists, other data partners, and GBIF, as I mentioned. Decision makers use these data when they're looking at maybe invasive species or rare species that are observed, new species to an area, that kind of thing. And it's a widely used program, so because of that, there's lots of amazing information available. So there's these stats were as of last night. So over 1 million people are part of it. There have been over 34 million observations and 270,000 species to date. So lots of information available to play around with, to look at yourself if you're interested, but also lots of information to contribute. So once again, this is just another way of looking at the 3 basic steps for our BioBlitz. The 3 basic steps are very easy. First, you have to sign up for an iNaturalist account. And it's free and it's very, very easy. If you want to use your smartphone or tablet, whatever you have, you can download the app specifically. And then basically you go take a walk, or you go in your backyard and you find something alive and you take a picture of it. And you submit it to iNaturalist. And I'm going to go through a step-by-step, but really it is that easy. And before I get into the step-by-step, just specifically thinking about the LLNF BioBlitz, because this is a geographically based BioBlitz where we're looking at the whole of Nantucket County, you don't have to specifically join the BioBlitz. If you make an observation on Nantucket during this time period, it automatically goes into our BioBlitz. And so in the map you can see that yellow cloud is our area of our project. So that includes Muskeget and Tuckernuck, Great Point, etc. And then all the red dots are observations that have been made so far. So one thing you might notice is there are no observations for Great Point, Coatue, and Cascada, if you're familiar with Nantucket. That's because those areas are currently closed to the public. Tuckernuck on the western side is more difficult to get to. And then I'm curious why, the South Shore by Tom Nevers hasn't been surveyed yet. So those might be some areas people are interested in going out to survey. So this is just another way of thinking before we get into the specifics of what motivates us, right? So not just our scientific questions of what's here, but what can be motivating about a BioBlitz and what can be fun about continuing with a BioBlitz. So one thing is to look at how many different species are here. So that's a lot of the competition factor of like who sees more species or how many species are on Nantucket at this time. But it's also fun to look at which are surprising and intriguing species. Birders love this all the time, like a weird variant that comes to the island, who's seen it first or what's an unusual, surprising find. Really great photos or videos are also fun to share. Species and taxa that are most common, so thinking about not just the rarities, but which are going to be the ones that people observe the most. That's an interesting kind of metric to look at. And then looking at the number of invasives versus the number of natives would be interesting. So these are all things that we're hoping to look at with the data after it's all in. So one of the things, these are some of the questions and some of the things that we can put out as the Linda Loring Nature Foundation at the end of the BioBlitz, kind of what are the metrics we're looking at, so what we're seeing. And then also the stages of life represented. So are we seeing adults, juveniles, eggs, larvae, and also the timing of all those things. So when are we seeing the eggs versus the juveniles versus the adults? So those are some of the questions we're looking at. All right, so now we're going to get into the how-to. So as I said, first take a photo of something living. So this was from, I forget, maybe a month ago when we first started as a family walking around. My son saw something green, the leaves are just unfolding. And so he took a picture with the app and then we uploaded it. And it could be just that simple. You can stop there. And I'm going to go into a more detailed version where we upload something, excuse me, and where it's a little more detailed, but you could stop there and then other people will identify that plant for you. This happened to be one of our shrub honeysuckles and it was just leafing out at the very end of March. So here is our kind of more detailed how something might work for you. I'm a plant ecologist. I have a huge affinity for turtles. And one of the things when we started the BioBlitz, thinking about, those those superlatives of who could be the first to see a turtle for the season, who could be the first to see a snake for this season. I was not the first of either of those things, but this was my first turtle of the year, and it happened to be on Easter Sunday. So we were taking a walk and we saw this little turtle in the side of the path in the leaf litter. It's that time of year where the turtles are all coming out, and it was a beautiful day. So I said, hey, look at that cute turtle. Let's put it on iNaturalist. So we opened up the phone, and this is what my phone would look like, my display if I open the app, the iNaturalist app on my phone. And then I click the observe button, which is the little camera at the very bottom of the app. And then when you click that camera, it takes a picture right through the app. So I have this picture is what I actually took and uploaded to iNaturalist. I said, yes, that's a good picture. It shows the side of the head, the leg, the coloring on the shell, so you can see for observation purposes or for identification purposes what you would need. And then you hit next. And then this is what you would see in the next page where there's still your little photo is at the top. And then you could share this right away if you just don't know what it is at all. You know it's a turtle, but you don't know what kind. You could just hit share. It already has the date, the time, it's geo-referenced to the location. But if you click on this part that says, what did you see? This is kind of one of my favorite parts about iNaturalist because it really helps you identify things. So you don't have to take pictures of things you know what they are. You could take pictures of things you just want to know what they are. So you click, what did you see? And the algorithm within iNaturalist uses your picture to figure out what it is. So you can see, based on when I said, what did you see? It says, we're pretty sure this is in the genus Chrysemys. Sorry, my Latin isn't great, but so we know it's a painted turtle. And so if I want to know more, I can click on the little I because I want some more information. And you can see that it says visually similar seen nearby. So that's helpful if you're trying to identify a species, knowing that it's been seen in the area before. So when I click on the info, this is one thing I love about iNaturalist, especially I know there are some teachers who are interested in using iNaturalist with their students. This is a great function because it produces a lot of natural history information about a species. So now I can read all the natural history information about painted turtles. And what you can't see is if you were actually on a phone, you could scroll down further and it would show you distribution maps of the species, and you can look at other like similar species and things. So it's a really helpful way for you to help identify. So if I click the select painted turtle, because I'm pretty sure it's painted turtle, then I click share, and that makes that observation live right away. And so then this is what my phone looks like where it says syncing. So if you aren't in a great Wi-Fi area, it will store that until you're in a Wi-Fi area. So if it doesn't sync right away, that's fine. You can go back to your home or your office or wherever you have internet, and it will upload later. And then sometime later, this is the other great part about iNaturalist, is how records are verified and identified. So even though I said, it's a painted turtle, other people who I don't know, who don't even live on Nantucket, who are just part of the iNaturalist community, identify it as an Eastern painted turtle. And so once there's agreement among multiple individuals, then it is considered research grade. So the last picture I have for this turtle is what the record looks like in iNaturalist. So this is off of my desktop computer. It's a little easier to see. Is you can see at the very top, it says painted turtle. It has a green flag that says research grade. So it's been confirmed by multiple individuals. And then on the right-hand side, you can see that there's a map of Nantucket with that turquoise dot is where this observation is from on April 12th. But the red dots all around it are other areas that same species has been found. So all those other red dots are painted turtles that were found on Nantucket. Not necessarily for the BioBlitz, because this is just a species record where it shows all the other painted turtle observations that have been made. So what I'm going to show you now are just some of the stats of where we are for right now. So this is our BioBlitz page on iNaturalist. So we have a BioBlitz project on iNaturalist. You can see the event is in progress. It's been going from March 15th to May 3rd. If you haven't heard the news, I'm sorry to say we just found out today that Massachusetts schools are canceled to the end of the year. So we might be extending it past May 3rd. We'll be talking about that, but usually, as I mentioned before, BioBlitzes are over a short period of time, and our period of time is getting longer, but I think it's useful for data and biodiversity exploration. Anyway, these stats that are on here from a few days ago, so we'll see what we have. But basically, you can see right on the top the number of observations we've had. Over 200. We've had 267 observations, more now since this was put up here. 128 species and 17 observers. So I know we have more observers now, and one of the reasons we're doing this talk is we want to really encourage more participation and more observers. If you click that stats button, we get even more information. And these are kind of the superlatives that are really fun to kind of play a little competition of who can see the most observations, the most species, and then my favorite is the most observed species. So which species do you think will be the most seen? And it's kind of funny because it's like, are they actually the most common species, or they're the species that are maybe most charismatic at any given time that people might be observing. So right now we have eastern red cedar, which is perfect for Nantucket, late spring, and then bearberry, another perfect spring, spring species as our most observed. We can break it down further, and we can look at, I love the color coding of the species information, because you can see what types of things people are interested in. So as a plant ecologist, I'm very excited that half the observations are plants. Of course, I made a lot of those observations, but it's like in the spring, people are excited about what's leafing out and what's starting to bloom, so people are excited about plants. We've had lots of insects, mollusks from people who are cruising our shoreline. We've got reptiles, amphibians, some birds, a few mammals. So it's really interesting to kind of see how that breaks down. And then we've had over 400 identifications. And so that means there's a lot of species that still need to be identified. Maybe they've been identified once, but you need at least 2 identifications to be research grade. So it's really interesting to see the observers and identifiers and how that works. So with that, I'm going to go over a few species that we've seen so far, and then also a little bit about what you can see on the iNaturalist website. So one of our most observed species is bearberry, which makes sense for Nantucket, especially because it is an evergreen. It's very common, so most of our walking trails in different parts of the island will have bearberry. And then also this photo is from April 14th, just last week, and it's already flowering. So you can see those really charismatic little pink bell flowers. So those are, the pollinators are going to be starting to get attracted to those very soon. So it's a highly charismatic species. And on the map, you can see, excuse me, you can see the number of other bearberry observations that have been made. So then this is an observation that I made April 5th, which was a white-tailed deer. And I put this in here because I want you to know that you don't have to observe the individual itself, especially for animals. You can see signs of animals. So you can take photos of scat, so fancy word for poop, for those who don't know. And you can take footprints. So this is an antler shed that I found when I was just walking, and the important thing was to take the photo where it was found. So I took this picture as I found it, because I didn't want to move it, because I think the location information is important. The map on the right that shows all the different location points for white-tailed deer, some of those might be actual deer observations, some of them might be scat or footprints and that kind of thing. Here is codium, which is also known as dead man's fingers. This was found at the end of March, walking along the shore. And this was just, I wanted to showcase that you can, things that wash up are also important too. It is a living thing. And the other thing to note about here that's different than other observations that we've seen so far is the little pink exclamation point at the top. So if you were to scroll over that, which I can't because this is a presentation, not actually online, is that indicates that it is a non-native species. And so you can if you didn't realize that, you could look at that it's non-native, and then you can look at the distribution of the species in a map, which is if you scrolled farther down, and you can see where the species is from. And so you can learn about the species that you're observing, not just what it is, but where it comes from, where else it might be found, and lots of other information. So here's a good example of something that you might not know what it is, and it might bring you down a rabbit hole of information. So this is something I love these species, and I tend to find these all over the place because my eye is drawn to them. And if you don't know what it is, it's like a little blob that you would see on a stick or a twig, and it is actually a praying mantis egg case. This one is the particular shape of it indicates that it's a Chinese mantis. But this is perfectly the type of thing that you might see in a walk and not know what it is, where there's a gall or like a weird thing on the twig or a leaf, and you're like, I don't even know what that is. Is it a fungus? Is it an insect thing? And you can take a picture of it and put it on iNaturalist and maybe learn more about it. So the great thing about this is you can see, first of all, that there's an exclamation point. So you know that Chinese mantises or mantids are not native to Nantucket. And this is a screenshot of, I recognize that it was a Chinese mantis, but then it was confirmed by a couple of other experts who suggested the ID. So I know, yeah, not only am I right, but now it's a research grade observation. You can scroll down further and learn about mantids, and so it'll show you pictures throughout its life stage. So even though you saw the egg case, now you can see what the adult individual looks like. And then the other bit of information you can get is on the right is the top observer. So this person on the leaderboard has seen 11 Chinese mantids. You can see when the last observation was, and this isn't just for Nantucket. When you get to the species information, it's for all observations. And the thing I want to point out is this figure, this graph on the bottom, and that is the seasonality. And so if you look at where the big spike is, basically August to October are when Chinese mantids are most frequently seen. And that's because they're huge. They're like 5 inches long. These are the ones that if you Google them, you'll see pictures of them eating birds. So if you saw one from anywhere from August to October, they're about this big. And so you would notice them and you would take a picture of it. And so that's, the biggest observations. Where we're seeing it between, March to April, it's pretty low numbers because they're still in egg cases. And then in June, there are no observations. And that's because the mantids have hatched. And when they hatch, they're super tiny. They're like 2 centimeters long. And so there's no eggs and you can't see the babies until they're bigger. So you can get a lot of information about the species you're observing by just kind of perusing through the iNaturalist website. And just a few more to show you. Nantucket shadbush, Amelanchier nantucketensis. So this is one I observed again. I didn't want to pick on other people's observations. So it's a terrible picture that I took, but I was excited to see it because it's the stick in the picture that has fuzzy buds on it. So some people say it looks like a pussy willow at this point because they're fuzzy little buds. But it's one of the first flowering plants on Nantucket, so in the wild. So I got excited that I saw it. And I knew it was a Nantucket shadbush. So you can see up top it says needs ID because there are very few people that will identify a Nantucket shadbush. So it's not research grade yet. The other thing you'll see is a VU, and actually I don't remember what the VU stands for, but here's a picture of what it will look like. So it's a much nicer picture than what I took. But if you look at the map on the right-hand side, this is an important piece of information. So no, I did not observe the Nantucket shadbush in the middle of the ocean. It is an obscured observation. So because this species is a species of special concern in some areas in its distribution, iNaturalist will automatically obscure the location information so you're not revealing rare species locations. So I think this is a really important thing for people to know because there are so many rare species on Nantucket that you don't have to worry about submitting information for rare flora and fauna and worry that you're revealing something. It will, it uses, excuse me, a few different databases that it draws from to know that this is a species of concern. And so now you can see that that big yellow box is like somewhere in here is where the Nantucketensis, the Amelanchier nantucketensis, the shadbush was found. So I think that's a really interesting piece of information for people to know. If you do want to purposely obscure location information, maybe it's at a sensitive site or a house, there is information on the iNaturalist website about how to do that. I'm not going to go over that today, but you can purposely obscure the information if you want to. And lastly, I just really wanted to highlight the bumblebee. So the reason I'm doing this is because, you know, you don't have to go on a far journey to find some bit of nature. So this is in my front yard. This is right by my front stoop. I planted a whole bunch of daffodils, so I'm not going to enter the daffodil because that's a planted individual. There are lots of daffodils you'll see in the BioBlitz, but that's because they're wild daffodils that have escaped cultivation, so they're in the woods. But this one is right outside my house. But what I did want to take is this bumblebee because it was the very first one I've seen all season. So April 16th, which happens to be my birthday, we were coming home from a walk and I saw this beautiful bumblebee and I hadn't seen any yet at all this season. So it was a first. It's been research grade verified as a two-spotted bumblebee. So that's exciting. So you don't have to go very far to find a bit of nature and wildlife for you to take to add to the BioBlitz. So I really hope that we can encourage people to join us. You don't have to join and take hundreds of observations. You can join us and make a few observations. We want to encourage as many people as possible to participate, not only because we want, you know, some of the biodiversity information, but we also want to share the findings with this group. Excuse me. We also are trying to encourage people to engage on our social media as well. We've been highlighting observations on our Facebook page primarily. We're going to be continuing to do that, highlighting kind of cool things that people have seen or maybe underappreciated species. We did the periwinkle a few weeks ago. And if you want to, if you're on Instagram, you can tag us with LLNF BioBlitz 2020. We're using that hashtag, and I'm going to, not just your biodiversity information, but I would love pictures of people making the observations, like your family out and about, or if you're a student, you know, like what you're learning, or just, you know, the more information we can share, we can kind of really keep it to physical distancing and not just socially distancing, because we want to kind of maintain those connections. So some additional resources to help you get started. If you go to our website, which is llnf.org, we have lots of information about the project. There is a PDF page that you can download and you can print if you want to. That is like a very explicit instruction, you know, where exactly, you know, that has a link to iNaturalist, how to set up the app, and how to move forward with the process. And so you're welcome to do that. Our Facebook page is where we put a lot of kind of day-to-day updates. And so I did this video at the end of March, which was my husband videotaped me doing an observation. It's totally ridiculous. It's freezing that day and it was really windy, but it's typical of what we're all doing right now, right? We're all getting out with our families and we're trying to maybe learn something along the way, teach our kids something. So this is a silly video, but it helps show basically how quick it is to take an observation. And then I would really encourage people who are interested to explore the iNaturalist website. There are so many amazing resources available. You can explore so much about different locations and different species, but there is a part of their website where they have a lot of short videos as tutorials. So I encourage everyone who's interested to at least watch the very first one, which is how to make an observation on a mobile device. They're short, they're very well edited, and there's good pictures. Probably much better than my super windy video. But if you're interested too, then there's other things like how to take great photos, how to take good identifiable photos for different species, and lots of different information on there. And the last few things, I just wanted to tell you about some other opportunities. There is a global social distancing BioBlitz that is going to be on May 3rd. We are actually one of the co-sponsors with this because the organizers, in starting to set up their BioBlitz, saw that what we were doing was very similar to what they were doing. So they encouraged us to be part of this. And so they had one in early April on a Sunday, and they had over 12,000 observations and 3,000 species. And that was a global effort. So they're going to do another one on May 3rd. And so for this one, you do have to select that you'd like to be part of the project. But what's great is if you are part of this project, any observation you make on May 3rd will go not only to the Linda Loring Nature Foundation BioBlitz, but it will be part of that BioBlitz as well. And then if you just haven't had enough iNaturalist and you need to hear more, it just so happens that this Friday, on Science Friday, for those of you who are NPR listeners, our local NPR station carries Science Friday afternoons. They are going to be discussing the City Nature Challenge, which is a BioBlitz that uses iNaturalist. And it's going to be from April 24th to 27th, that BioBlitz. And it's usually a competition between cities, and it gets really heated about who can find the most species. This year, because of stay-at-home orders, there's not going to be a competition, but they're going to be specifically talking about the City Nature Challenge using iNaturalist. So there'll be a lot more information. I'm excited to hear it, to see what else they have to say about iNaturalist. And with that, I want to thank everybody for joining us today. I would love for people to join in and start to take some more observations. You know, it's a great way for those of us who are educating our kids, and it's a great way to have a science lesson, to learn about the species if you don't already. This is my son giving a peace sign under some eastern tent caterpillars that were hatching last year because he knows how much I love caterpillars. So, you know, it's a way that we can bring our kids out and teach them something. And tomorrow's the 50th anniversary of Earth Day. So what better way to celebrate than to get outside and, you know, look at some biodiversity. And with that, I will take questions, but I have one more slide before questions, which was just to say, excuse me, that our next speaker next week is going to be, our next virtual science pub is Sam Kefferstan from Mass Audubon's Nantucket Sanctuaries Director. He's going to be talking about the Coastal Waterbird Program for Mass Audubon, and I'm really excited. And for those of you who are on Nantucket, Sam is new to the island this year, so it'll be one of the first times we get to hear him talk about the programming at Mass Audubon and yeah, and what his plans are. So once again, happy to take questions, and thanks for being here. All right. I don't know if Seth's coming back.
Yeah, hi Sarah.
Okay, hi.
Stop sharing your screen and I can fill the questions, or you want
Got it. Stop sharing. Okay.
Hi everybody. We can all give a round of applause to that excellent presentation. I had a few things that came up in the chat. Let me just pull them up here. One of the, and then we'll get to the question and answer. One of the things that came up in the chat was that one of our attendees commented that the VU means that the species is vulnerable to population declines.
Thank you.
And we also had a question from a participant who had recorded 2 photos at Sanford Farm. One was a distant photo of pines that was identified the next day, and one was a close-up of a red twiggy grassy plant not identified. They are looking for guidance on how to better take photos for grassy or twiggy plants.
That's a really good question, and I can definitely answer it. But I also recommend looking at some of the iNaturalist suggestions. One thing that it took me a while to realize on iNaturalist was that I could take multiple photos. So especially for something big, like even a shrub, because people always send me pictures of plants and want me to identify them, and it helps to have both a close-up of like a leaf. I mean, this is specific to plants, but you know, a leaf and the close-up to help you get the identifying characteristics, but then a step back to look at the whole individual, maybe, you know, a scope of how tall, how big it is, and maybe a little bit of the habitat. So those are two things for kind of bigger things. For smaller things like grasses, sometimes it helps to put something behind. So a lot of times I'll put my hand, or if I have a piece of paper, something behind, you know, behind it, so that you can focus on that small thing, or, you know, thin twiggy thing. And sometimes adding a few different photos would also help with that, because if you take things very close and then make it maybe a little bit farther back, so that you can kind of see, you know, a few different angles of it is really helpful. The other thing with identifying things is sometimes people don't get to it immediately. So I have found, you know, you get a notification if any of your observations have been like commented on. So you get, you know, like an email or whatever. And it's funny because I will get comments that something I took a picture of last summer was identified. So, you know, certain things don't get identified right away. We are trying, as the Linda Loring Nature Foundation, since this is our BioBlitz, I'm trying to go through a lot of the photos and identify what I can. But like I said, for research grade, you need at least 2 observations. So, you know, or sorry, 2 confirmed IDs. So, you know, some of it is just it takes a little bit of time. And I would remind people too, it's kind of funny, if we did a BioBlitz the way we normally do programming at Linda Loring, where we planned it well in advance, and we had a group of people that decided we were going to be doing this, and we had this, you know, long lead up to planning it. A lot of times you have multiple experts on hand to identify things as soon as possible, especially for like insects or really specialty groups. But because this is sort of, you know, in response to us in staying at home, and, you know, the social distancing orders, you know, we're a little bit reliant on the community of iNaturalist identifiers, more so than I think we would be normally. So it's just a little bit of time on that.
Thank you, Sarah. Moving on to the next question, we have one from Doug Horst. He's asking, if an observation is made, but a picture cannot be taken, the thing moves away, or there's too little light, can the observation be recorded in iNaturalist in some way?
That's a good question. So we have done that before, and you can enter the species information. It's a little more labor intensive, and I don't know if you can do it from a phone. I know that we did it over like on a regular desktop computer before. It won't be recorded as research grade, because there's no way for anyone else to support that verification. It's not that it's not a worthy thing to enter, it just won't ever get to that level of, like, because no one else can say they can't see it to verify it. The reason we did it is, you know, I think we did it last summer at the Linda Loring Nature Foundation. We keep track of the biodiversity of the property on 110 Eel Point Road, and we had a species list of pollinators from a research project, and we entered all those just so they were all in the same database. And similarly, we didn't have photos of those individuals, so it's possible. It just won't be research grade.
There's also some questions about, is it possible to upload audio recordings like bird songs for ID?
Yep.
And Scott Smyers in the comments is saying that you can do that.
Yeah, you can. A lot of people do. I know Scott is a herpetologist, so he does the spring keepers, the frogs, and insect sounds as well. So yeah, you can record the sounds. It's a little bit different data entry wise. So I would suggest recording them however you prefer to record, whether, you know, using your phone most likely, and then uploading it later, because it's not as simple as when you are on the phone and like what I showed where you just take a picture.
And I believe you can record a video as well. Is that correct?
Yes. Yep. You can do videos as well.
Thank you. We have a question from Jeannie Garal. Do you have a policy on removing invasives once identified?
That's great. Hi, Jeannie. Good to see you, or not see you, hear from you. We don't currently have a policy. I think it's a property by property policy. At the Linda Loring Nature Foundation, we do, where if, you know, we're pretty strict about, because we haven't had so many invasives, that as invasives are found, we try to remove them right away. We've had that with the shrub honeysuckles. We're really trying to get those as the, you know, they're kind of spread around. But being that this is an island-wide BioBlitz and we don't have the property rights all over everywhere, I would be hesitant, but it would be really interesting to see if any new invasives come up, because we're starting to record more information. In Massachusetts, I know Jeannie's not in Massachusetts. In Massachusetts, people use EdMaps, which is an invasive species database, to record invasive species information, and that's what the state looks at for new records. So if there was a new invasive that came out of people kind of canvassing the island and looking for it, we'd definitely be interested in finding that out. So I think that's going to be like a stay tuned also. One thing I was I know that there are a few more questions, but I did want to mention that I don't know what the end of this is going to look like. You know, this is all new and things keep getting extended, but what I'm hoping is that at the end of this, we can have some kind of, you know, both we'll summarize what we find and have some statistics to share with what we've seen, invasives versus natives, what's new and surprising, and some of those questions that I had put on the presentation. But also it'd be great to have a get together too, where people can actually do some kind of group walk or information at some point too. I don't know what that would look like, but it would be interesting to see, you know, what the data show and what the observations are.
Great. The only other question I'm seeing right now, this might be relevant for the people who watched last time. Do you have a special cocktail recipe to suggest while people follow along with your talk?
I know. So I did not think of a cocktail recipe for this one. So usually at Science Pub, for those who are new to it, because we're usually at a bar, we you know, it's a way for people to be kind of comfortable and relaxed, and, you know, feel comfortable to ask questions. And I failed to have a cocktail recipe because I have water. I have our Gray Lady pint glass, and so I would say that, being that we're on Nantucket and it's starting to get grayer and grayer today, a Gray Lady would be an excellent beer to toast with my talk today.
We just had one question come in here from Sam Keperstan. Hi friends, awesome presentation and thanks for the introduction for next week. My question is, do you have go-to resources to further research beyond iNaturalist once you have an ID, particularly for plants? So once you've gotten what the idea of that is, where would you go next?
Yeah, so that's a great question, Sam. So my go-to right now for plants, especially for online resources, would be GoBotany. So GoBotany is a New England website hosted by the Native Plant Society, which is formerly the New England Wildflower Society. And so GoBotany is a great online resource that has wonderful pictures of plants, and it also has great, easy to use keys if you have a plant and you have just so much information. So if anyone's interested more in GoBotany and about that, you know, feel free to email me. The other thing for if you're not into using the online resource, GoBotany is a companion to the Flora Nova Anglii, which is the very more dense and rigorous plant key. So it's a very big book without picture, mostly without pictures. So, but that's kind of the New England based resource that I use. And then I'm happy to, talk. Field guides is like one of my favorite things. So there's lots of great field guides, but I'd say Go Botany is kind of the standard.
Thanks,
and
Sarah.
then you get to say Go Botany.
Sarah, I have one question. I'm not seeing a ton else come in, but my question is related to the option to check captive or cultivated species on iNaturalist. I know in your presentation, you said we're really trying to get people to go out in nature and find things that aren't planted. But if people, for whatever reason, can't really leave their house or don't feel comfortable, can they still just observe things that are in their yard that they know they planted if they check that box?
Yeah, if you check that box, you can. It's just a way for scientists to know that are using the data that it's a planted individual. And there is, scientific benefit to that too, because you're showing that it can grow there. I think that's, it depends. And by checking that box, you're giving, what we call metadata. So it's data about the data. So you're saying that if a scientist later wants to use the data for a certain type of species being around, then they know, well, it was planted, but then it's up to the scientists to decide whether or not they want to use that. So as long as you're giving that information, if you're not going to check the box that it's been cultivated, I forget what the box says, it's like cultivated and
Captive.
Thank you. Then if you're not going to check it, then I would encourage you not to record those species that are, clearly part of the are planted.
Thanks. It doesn't seem like we have any other questions. We have lots of comments here in the chat that people miss you, that you did a great job, and that it was very
thanks.
So again, let's thank Sarah Bois, Director of Research and Education at the Linda Loring Nature Foundation, for her awesome presentation today for Virtual Science Pub. And like she said, Sam Keverston will be speaking next Monday, April 27th at 4 p.m. We'll be posting the link for that. So stay tuned on our website and Facebook to get the joining information for that program.
Thank you, Seth. And thanks, everybody.
Thank you.
Have a good weekend.